right, we're still in the reproductive system, just like we're still in winter, apparently. It's just endless. So we'll forge on, and then I'm going to try and get home real quick today. Physiology, we'll start with the sexual response in the male. First thing we need to talk about is erection of the penis. This is facilitated by parasympathetic arousal from the autonomic division of the nervous system, parasympathetic stimulation from the central nervous system. So the rest and digest division, right? You remember that probably? Yes? That has to be active to be able to achieve an erection. That's why stress that gets you into the more sympathetic part of the spectrum might interfere with erection of the penis. So it needs to be a fairly relaxed situation. There's release of nitric oxide locally, and that leads to relaxation of the vessels entering the erectile bodies. The three erectile tissues get increased blood supply, and they fill up with blood, and that compresses the drainage veins. So there's a period of time when more blood is going into the erectile bodies than out. So compression of drainage veins. By the erectile bodies themselves, by the engorged tissue. And with increasing stimulation, though, we get a switch from parasympathetic input to sympathetic input. So ejaculation during the male orgasm, sympathetic stimulation. It's a sympathetic spinal reflex. And that leads to contraction of the duct system, particularly the vas deferens or ductus deferens, as well as the seminal vesicles, the prostate, and semen gets propelled along the ductus deferens and urethra then, and there's a constriction of the urinary bladder sphincter, since you do have the co-option of the urethra by the reproductive system. The bladder sphincter constricts, heart rate gets elevated, and blood pressure is elevated as well. And after ejaculation, there's a lag time or a latent time, whatever you want to call it, where a male cannot achieve another erection and ejaculation right away. And these lag times increase with age in men, so that gets more and more spaced out the older you get. In the female, it's somewhat similar. In the female sexual response, we don't talk about erection, even though the clitoris gets engorged and the labia do get engorged. But in females, it's called the excitement phase. And just like in the male, this is under parasympathetic control, rest and digest. And that leads to the engorged clitoris. We learned last time that's basically the same as the penis. So you have increased blood flow to the vaginal lining, even the breasts. And then we have also vestibular gland, mucus for facilitating intercourse for lubrication. So this is all the parasympathetic excitement phase, and then just like in the male, orgasm is the switch to the sympathetic part of the autonomic nervous system. Females, the orgasm is muscle tension overall, elevated heart rate, blood pressure, rhythmic contractions of the uterus actually, and that is thought to sort of suck sperm up potentially into the cervix of the uterus, and then the elevated heart rate and blood pressure. And then bonus, no latent period. So multiple orgasms in females are possible without a waiting period. Okay. Now, sex is fun to make babies, more likely, right? So we are going to now look at, before we get into the fertilization part, we'll look at the production of the gametes, the sperm and egg that have to come together to form a new human being. And this is a picture that's no longer in your book. I guess it was just too controversial. I always like to still show it because it just speaks a lot about the politics in this country. When I came here 10 years ago, I don't know what edition of the book that was at the time, but this was in the book. And you may note that she's African American, he's white, right? So the next edition, suddenly, oh, how funny, they were both African Americans, they were both black. So somebody must have written the publisher, this is not okay, this interracial business, take care of it. So they fixed it, right? And then I guess even that was too controversial, so since then it's been gone. It's never reappeared again. Isn't that something? This is like 2013. Unbelievable to me, anyway. But it's a useful figure because it just kind of summarizes the life cycle. And has the terms on there that we'll need to look at now. So we're going to look at gamete production, eggs, sperm, how do you produce them, and they get produced by each individual in the process called meiosis, and that's going to be a little different from the regular cell division that we've talked about before, the mitosis. Right, so in AMP1, relatively early on, we go over regular cell division, mitosis. You learn all about that prophase, metaphase, anaphase, telophase, that stuff. So now we'll look at a modified process that will have the similar names of the phases, but it's called meiosis. And the process is different because when you produce sperm and eggs, you have to make sure each one of those only has half the genetic material in it compared to a regular body cell. So when you do fertilization, you don't double the DNA content every generation, right? So in meiosis, we're taking body cells and basically having, splitting up the genetic content in there, so that sperm and eggs only have half the complement of chromosomes. If you only have one set of chromosomes, that is called haploid. So gametes are haploid, and that's indicated with the small n. Haploid gametes only have one copy of each of our 23 chromosomes. All of the other body cells have two copies, and that is called diploid or 2n. So you have these two haploid gametes, fertilization brings all the chromosomes back together, and you have a diploid potential new organism, the zygote, the fertilized egg. And then that fertilized egg undergoes regular cell division, mitotic divisions, to grow into an embryo, potentially a fetus, and so on. And in adults, we're still diploid with our 46 chromosomes, 23 pairs. So we need to look at the process of meiosis. So hopefully this is kind of going to be review. So you remember we have the 23 pairs of chromosomes. This is from chapter 29, I guess, a karyotype of a male. So we had 23 pairs of chromosomes in humans, and the chromosomes are homologous to each other, or they're called the homologs. So here are the two homologs of chromosome 10, here are the two homologs of chromosome 9, and so on. So just kind of reminding you of that terminology, the homologs. And 22 of those pairs are autosomes, and then we had that one pair of sex chromosomes. You remember this stuff, I assume, at least vaguely, right? Yes? No? So the sex chromosomes were XY, male, and here you see the X and the Y chromosome, so this is why this is a karyotype of a male. The Y is much smaller. It only has the genes to turn the plumbing into the male that we talked about last time. And XX is female, and so having chromosomes in pairs is called diploid. And you hopefully remember that one set of your chromosomes came from mom and the other set came from dad. So when your mom's egg and your dad's sperm combined, you made another diploid zygote that then grew eventually into you. And remember that the father determines the sex of the offspring, so sperm has either an X or a Y chromosome in it. So in mammals, the male is the sex that determines the sex of the offspring. Okay, so everybody is back on the same page with this stuff now, right? So pairs, chromosomes, and we understand the need to get rid of one half of that stuff, so when you combine sperm and egg, you don't have extras. Now, whoops. So here's that again. Now, before I go into meiosis, I just want to go over regular cell division, mitotic cell division again, just to, to just sort of resurrect some of that, what we've done there in ANP1. And this is in your chapter 3. It's since been updated, so you can just use your figure 333 for that. It's the same information is still on this. So we had interphase. During interphase, DNA replication occurs and the cell grows, and when we are nearing irregular cell division, you get the early indication of a spindle getting set up by division of the centrioles. And then we enter the first phase of mitosis, which is prophase. Both of these are prophase pictures. And several things happen there. The nucleus breaks down, so here still a nuclear envelope, and there's disappearing. The chromosomes are condensing so they can experience the rearrangements and being physically pulled around without breaking. And the spindle with the microtubules is setting up. And the microtubules will, sometime during prophase, attach to these chromosomes and start pulling on them, and eventually the microtubules will attach from both ends on the chromosome in some area, and their pulling will eventually line them up, and that's when you see something like this, and that's when you know you're in metaphase, when the chromosomes are sort of equidistant from the poles of the cell. The spindle is complete. Then during anaphase, the two copies of the chromosomes pull apart. It's called separation of sister chromatids, the exact copies. And then in telophase, you pinch them off and you reform everything. And you have two identical daughter cells. You kind of remember this, right? So you duplicate the DNA before you do anything, and then during mitosis, you pull apart those duplicates, and you have two cells that have exactly the same genetic content as the mother cell. Now, for meiosis, we have to get to reduction, right? So you need to have fewer chromosomes in there. Instead of 46, you need only 23. So let's lead into that. Meiosis. First, we'll define it a little bit. There are different definitions for it, like anything. I'll just use this, the process of cell division that results in the production of 6 cells, sperm and eggs. And then just to be painfully clear, right, so your body cells have 23 pairs of chromosomes, and the 6 cells, sperm and eggs, have half the chromosomes, 23, and not just randomly, just half, but one member of each pair only. Okay, ready? Now you may think, pretty much everybody thinks that when they first hear about it, well, the easiest thing to get to half of the chromosomes is simply eliminating the step of DNA replication, right? So you don't replicate the DNA, you line them up, pull them apart, one member of each pair into the daughter cells, and you're done. But that's not what happens. What you're actually seeing in sperm and egg production by meiosis is you do replicate all the DNA beforehand, and then you divide everything twice to finally get to half the number of chromosomes. So the DNA replication technically takes you to twice the number of chromosomes, and then you halve them, brings you back down to 46, have them again, that brings you down to 23 finally. So we have 2 rounds of division, and they're called meiosis 1 and meiosis 2. And here's figure 27.7 showing meiosis 1.
Bless you.
So just like in mitotic cell division, mitosis, you're going to replicate all the DNA during interphase, and you have the 2 copies very closely attached to each other, but they are replicating. So interphase events, as in mitosis, meiosis is preceded by DNA replication. Then we'll enter prophase of meiosis 1. Luckily, the phase names are the same, in the same order. So we have prophase 1. Some stuff happens there that's very similar to mitosis. The nuclear envelope breaks down, the spindle starts to assemble, those microtubules come out. But there are also some differences between this and mitosis. The big difference here in prophase 1 is that chromosomes actually come together as pairs. So when you look at the example, this is an organism with only 2 pairs of chromosomes for simplicity instead of 23 pairs, right? So that would make it really too busy. So we have a hypothetical organism with chromosome 1, it's 2 copies, chromosome 2, it's 2 copies. You see how each chromosome has like 2 little things? Those are the duplicates that we made in interphase. Okay, are you still following? All right, so we have, each chromosome has been duplicated, so there, if this is the big one, let's say that's chromosome 1, the purple member of that has already duplicated, the yellowish member has already duplicated before. And they come together in as chromosome pairs, and they will actually quite often exchange genetic material. So where pieces break off from one arm and reattach to the other, where they literally switch out genetic material. Here in this metaphase picture, you can kind of make out that this purple guy has a little bit of yellow on it, and the yellow guy has a little bit of purple on it, right? So this is totally new. This is not something that happens during mitosis, only during meiosis. It's called crossing over, crossover, right there. Where do they describe it? Okay. Additionally, synapses, that's what they call it when they're lining up. Homologous chromosomes come together along their length. During synapses, the arms of homologous chromatids wrap around each other, forming several crossovers, literally just crossing shapes. And they trade segments at points of crossover. Crossover is followed through the diagrams below. So where you see bits of the wrong color kind of attached to the chromosome arms. Okay, so this is new. Crossing over, stuff breaks off and reattaches on the other chromosome. Okay, so write that one down. It's one big difference. Then we have metaphase when stuff lines up. During metaphase 1, though, instead of everybody lining up kind of in a row, here we're lining up as pairs. So there is another difference. Metaphase 1, the chromosomes line up in pairs, not in individuals. Metaphase 1, big difference between meiosis 1 and mitosis is the chromosome line up in pairs now. During anaphase, things get pulled apart, but what we're pulling apart during anaphase 1 is the chromosomes, the homologous chromosomes. So here we have separation of homologs, not of sister chromatids yet. So during anaphase 1, we separate the members of homologous pairs, separation of homologs. So if chromosome 1, one goes this way, one goes the other. Chromosome 2, one this way, one the other, including little bits that they broke off and exchanged. And then during telophase, we pinch them off into 2 new cells. Okay, so you have 2 daughter cells. Now, we'll continue the process. These 2 daughter cells then enter meiosis II. Here are the 2 daughter cells. They'll enter prophase II, so no nuclear envelope condensation of the chromosomes. Now there's no more crossing over. So we progress to metaphase. Metaphase now looks a lot more like what you saw in mitosis. They're lined up in a row in metaphase 2. And then in anaphase 2, now we're finally separating sister chromatids. So the 2 copies that you duplicated during interphase are being pulled apart. During anaphase 2, you get the separation of the sister chromatids, and then telophase, you just form the daughter cells, and at the end we have 4 daughter cells from that initial mother cell. In regular mitosis, we only had 2 daughter cells. Here we have, after 2 rounds of division, meiosis 1 and meiosis 2, you have 4 daughter cells, all shown here at the bottom. Okay, so there were quite a few differences. Do you want me to go over that one more time? Just make sure you got all the differences for each. Yes or no? Just move on. You want to get out of here or go over it one more time? Nobody cares. Well, let me go over it one more time. I think it's just... So we duplicate the DNA, just like in mitosis during interphase. Then we enter the first round of division, meiosis 1. It has all the same phases, prophase, metaphase, anaphase, telophase. Something new during prophase 1, the crossing over, because the chromosomes line up as pairs. Pieces break off, reattach, and you get different combinations of genetic material. So that's big deal about prophase 1, this crossing over. Big deal about metaphase 1, lining up as pairs, not as individuals. Big deal about anaphase 1, we're separating the homologs, not the duplicate copies. Separation of homologs, telophase, unremarkable, pinching it off, and those cells progress into meiosis 2. So here are the 2 daughter cells at the end of meiosis 1. Prophase, nothing remarkable there. Metaphase 2, now they line up in a line instead of as pairs, because the pairs have been separated. Anaphase 2, now we're finally separating the sister chromatids that we generated during interphase. Telophase, you pinch off the cells, and you have a total of 4 daughter cells at the end of meiosis, where mitosis only generated 2 daughter cells. Each of those 4 daughter cells of meiosis are haploid. They only have 1 copy of each chromosome. So in these simplified examples, there's 1 copy of chromosome 1, 1 copy of chromosome 2, and we're done. So in humans, you'd have 1 copy of each of the 23 chromosomes. Here is a comparison that you can look at that sort of highlights more what the differences are. 27:6, so it has mitosis on one side and meiosis on the other. So I really recommend you go look at that as you're studying this, because it's just a different way of showing that again. So now what we'll do is look at how this process actually occurs inside the testes and ovary. Okay, and we'll start by looking at the male, because it very much follows this pattern. You have one mother cell giving rise to 4 daughter cells, which then mature into viable sperm, hopefully. And then after that, we'll look at what happens in the female, which modifies this process quite a bit in the timing and what happens to the cell. So meiosis in general. Now we'll look at male, which pretty much is this right there. Oh, this continues on. Sorry. There it is, male. And the production of gametes in the male testis is called spermatogenesis. Spermatogenesis. Spermatogenesis, making sperm cells by meiosis, regular meiosis, 2 rounds of division. All of this occurs inside the seminiferous tubules of the testis. So inside the testis are all these tubules called seminiferous tubules. And it happens from the wall, like if you have a cross-section through a tubule, the oldest cells are near the baseline, and then as they mature, they move towards the center where the opening is going to be, and fairly mature sperm will be released into that opening. So spermatogenesis. So what you see on this 27-8 is just right up there, they show a little bit of the wall of the seminiferous tubule. So this would be like gigantic, right? So if you were to show the whole thing, it would be huge. So this is the wall of the tubule. The lumen would be towards the bottom of the figure. Now in males, males are always full with a supply of stem cells for producing sperm. We looked at how many are released with even just one ejaculation, up to 130, 140 million per milliliter. So lots of sperm are produced all the time in males after puberty. And so you have a supply of stem cells. So very close to the wall of the tubule, you have these stem cells. The stem cells are called spermatogonium. Okay, so know that name, spermatogonium. It's the stem cell of sperm production. And they undergo regular cell division to always have a supply of spermatogonial cells. So you see here the stem cell, spermatogonium, undergoing mitotic regular cell division, mitosis, to produce daughter cells. And then only one of those daughter cells will progress and actually enter meiosis, the cell division, to produce sperm. So here we have a daughter cell of this stem cell right here, and it will enter meiosis 1 and starting to move away from the edge. It's starting to make its way closer to the lumen, and the cell that really enters meiosis 1 is this primary spermatocyte. So this guy is really committed to undergo cell division to produce sperm. It completes meiosis 1 to produce 2 secondary spermatocytes, and then the secondary spermatocytes undergo meiosis 2 to produce 4 daughter cells, and these 4 daughter cells are called spermatids. Okay, so the 2 rounds of cell division of meiosis, the primary spermatocytes, round 1, produces 2 secondary spermatocytes, round 2 produces 4 spermatids. Okay, so that whole meiosis, 2 rounds of division, primary spermatocytes makes 2 secondary spermatocytes, each one of those makes 2 more spermatids during meiosis 2. And all of those spermatids are equivalent, and they're little round cells, so they're not functioning sperm yet, but they have the right amount of genetic material. They're haploid, and they only have one copy of each of our 23 chromosomes. Okay, so again, the primary spermatocytes start meiosis 1, finish it, then they're called secondary spermatocytes. They'll start meiosis 2, finish it, and they're called spermatids. So you get one mother cell giving rise to 4 daughter cells in sperm production. And then these spermatids are not able to fertilize anything yet. They need a tail, they need a little factory to propel that tail. So the spermatids undergo a maturation process that is called spermiogenesis, and we'll look at that one as well. So we're at the end of meiosis, we're not already having fully functioning sperm. These spermatids are finished with meiosis, but they're not sperm. This is what it looks like a little bit more realistically, I guess. Although if you would actually look at a slide, it would still not look like this. So they're trying to show it a little bit more. So here's the edge of the tubule, and as the cells divide, they move towards the lumen of the tubule. Okay, so this is this other part of that same figure. Labeling the cells, it's not that meaningful. They all kind of look the same. The sizes are different. Okay. So I just wanted to show that for interest. So this maturation to actually make functioning sperm is illustrated here on 27.9. 27.9. Spermiogenesis, right? So make sure you get the different. Spermatogenesis was the meiosis, where you take the one mother cell and make 4 spermatids. Then they mature by spermiogenesis into actual spermatozoa. Look how long that takes, 24 days. It's quite a long process. Important steps during spermiogenesis is activating the genetic material in the sperm to produce this long tail. The tail is a flagellum, so it's motile. It can use ATP to produce motion. So we grow a flagellum. Flagella are supported by microtubules, so that's growing out. Then to power that flagellum, you need a lot of ATP, so we aggregate the mitochondria around the base of the flagellum. So here they're lining up in what will become the midpiece of the sperm. So we need all the mitochondria to make the ATP to power the flagellum. And then we also form a very important structure for fertilization that will come to lay at the first part of the head of the sperm, covering the nucleus, and that's called the acrosome. And we'll look at the function of that when we look at fertilization. But the acrosome is a vesicle, a membrane-bound sac that is at the very front end of the sperm, and it contains important enzyme that we need for fertilization. So that needs to be set up as well. Once you have your mitochondria, your flagellum, and your acrosome, the rest of the cytoplasm, the rest of the organelles are going to be stripped away. To make that's mostly to make the sperm really light and easy to propel. It takes less energy to propel a small sperm compared to a large one. So we're stripping away most of the cytoplasm. So a finished spermatozoon looks like this. Head has the nucleus, the acrosome, midpiece has the mitochondria, and the tail is this long mobile flagellum that propels everything along. And is this correct? Okay, so I have a little bit of time, so let me just finish up this kind of a nice etude with the hormonal regulation in males. This looks really crazy, but we're not going to deal with too much of the issues Everything starts in the brain, and you'll see it's going to be the same in the female. So this is figure 2710, hormonal regulation of sperm production. And it starts in the hypothalamus with gonadotropin releasing hormone. If you remember that stuff from the beginnings of this course, the releasing hormones from the hypothalamus go to the anterior pituitary, trigger the release of stuff, right? Remember that part? Releasing hormones coming down through that little system. Remember that hypophyseal portal system? And the hormones that stimulate the testes to produce basically testosterone are the gonadotropins. So FSH is follicle stimulating hormone and LH is luteinizing hormone. Both of those names expressly relate to their function in women because that's where they were really described first. What is the function of these hormones? How do eggs ripen in women? Now we know that the same hormones are also active in males. For example, if a male does not have sufficient FSH, he's not going to be fertile. Okay, so you do need this in males as well. FSH stimulates this thing, the androgen binding protein, which basically keeps testosterone around in the testis. Keeps testosterone around. Androgen binding, the androgen it binds is testosterone, and testosterone itself is stimulated by LH, and it comes from the interstitial cells, so testosterone is being released. So we need these hormones with the female-sounding names in males to stimulate testosterone release and then to keep that testosterone around in the seminiferous tubules where it stimulates and maintains spermatogenesis, meiosis in the male, and maturation of the sperm. So they're not unimportant in males. The release is kind of relatively level once you reach adult age. You'll see in the females there's actually a really crazy sort of up and down of FSH and LH when we'll get to that. So once you're an adult in males, it is fairly level, but some crazy stuff goes on in males as well. Sometimes some of it before birth. This is the red line is testosterone level. Birth right here. So look at this. Probably having to do with setting up the male anatomy, basically. And there's also a definite masculinization of the brain. There are noticeable differences in the brains that result from testosterone exposure before birth. So you get that spike here, then you get a drop off, and then there's another spike early on in baby boys. I have no idea what that does, but then it's sort of low, and then it obviously starts puberty. That's when the testes become active, and then during adult age, testosterone levels and everything stays pretty much the same. And in women, you'll see every month it's going to go up and down. So we'll leave it at this. And next time we will compare what happens in women with egg production, which will have quite a few differences. So make sure to kind of look over the sperm production before Wednesday, so that makes more sense to you.